Good afternoon. Welcome back from a coffee break. My name is Andreas Gisel. I'm a senior researcher at the Italian Institute or Italian Research Council. And the institute I'm working is this Institute of Biomedical Technologies. I'm collaborating with IITA, and that's the reason why I'm talking about Africa, and I will explain why. And since I'm a bioinformatician in biomedicine, I need some collaborations with plant people because in Africa, I'm working with plants. So before I start, I want to show you this ball over there, is World MNet. Just want to lose 2 words also for this. I'm member since 2003 of MNet, and all except of one project I got through MNet. So collaborating with members of MNet, I have had the opportunity, or still have, to be in projects via MNet. So just to tell you, networking is the key for your career and to get money for doing research. So a couple of years ago, I moved with my family to Africa to live and work there as a bioinformatician. And didn't go. What is not moving? Okay, I'm not moving to this, or I was not moving to this Africa. I was moving to this Africa. So unfortunately, in Africa, there are still countries, we call them developed or developing countries, which are not developing. There are people, half of people living with less than 1.5 euro per day. And when Eric is saying at the beginning in the first talk, diet is your health. What is your health if you don't have food? So I was working, and I'm still collaborating with the International Institute of Tropical Agriculture in Nigeria. You see here Africa and the places they are involved. The red points are the hubs. So a hub for West Africa, Ibadan. Here is Kinshasa, is Central Africa. Dar es Salaam is in Tanzania for East Africa. And here we have Lukasa in Zambia for South Africa. This is the campus I was living, so I'm not really living in these places you saw before. I don't know where I would have survived that. You have regions over here where we had the labs and the offices. Here in the center, you see the places where we lived. Then you have all the experimental fields, a lake, and the rainforest in the back. All this belongs to this campus of IITA in Ibadan. So I was working, or I'm still working as a bioinformatician in collaboration with the breeders. That's the main activity of this institute, to breed for better varieties in different crops. The crops I am involved is cassava, and I will introduce this to you because I will show you examples of cassava. Yam, this is another tuber plant typical for Africa. Maize and cowpea. I will concentrate on this, otherwise I would talk about 3 or 4 hours. So it's cassava or topiaco or manioca. There are different names. Manihot esculenza crans is a tree, is Euphorbacea, and is a major fruit crop for more than 800,000 people all over the world. Most of them is produced and eaten in Africa. If you compare actually the amount of cassava which is produced with the potato, you see how much distributed cassava is. And the most important part is this one here, is the highest food energy you can produce on land. It's almost double of rice or wheat. It's a very important crop. It is also important because it is quite drought and heat resistant. So it is facing climate change. And that's the reason why more and more countries, including China, are pushing hard in the research of cassava. It yields also in low quality soil. So if you go to the place like I was in Ibadan, outside the beautiful campus, what we call the Golden Cage, you see that behind of the houses, they grew cassava. Because this is the stock in case they don't have food, they go and pull the plant behind the house and have a dinner with cassava root. So it is also very, for keywords like circular economy, it's very interesting because you use the whole plant. What I was talking about is the root. So most of people eat the root, which in most cases is converted in flour and then produce all the food people are eating. The stem we use for propagating, so it's a clonally propagated plant. And the leaves can be either food, some are eating these leaves, or can be feed for animals. The peels of the roots also become food for animals. So you see the whole plant gets used as a plant, a crop. The other important part actually, which we started to plant and experiment with cassava in Italy, is gluten-free and has a low glycemic index. So it is interesting food product for these people which have this problem. So my study 10 years ago when I started was in a big project where we wanted to understand the sink-source relation between the leaves and the root. The interesting part of cassava is that the root is an intermediate storage. So if the plant needs energy, it will pull out the energy again from the root to produce leaves or stems. But the study should have, or it was, on this topic. But we realized that the most simple question, when and how these storage roots start forming, nobody knew. So me, as a bioinformatician with my team, we went to the field and started to dig out roots to see when these roots, the storage root starts to develop. This is a plant, you see the stick, which you stick into the soil. On one side, the side in the soil starts to root. On the other side, you have the sprout. And we wanted to know which root of these and when they start to bulk and store starch. Looking at these roots, we had kind of two types, what we call potential storage roots and potential fibrous roots. And we did a very simple experiment. We opened the soil, we labeled with rubber bands with two different colors, either what we think is this, potential storage or potential fibrous roots. And the interesting result is that none of these ones, what we claim should be fibrous roots, were bulking, whereas this one here, they started to bulk. So now bioinformatics starts. How can I differentiate between these roots? We sampled over time storage and fibrous root samples and run RNA-seq data analysis. And here you see after processing, I don't go into this, I guess, is basic NGS bioinformatics. We found genes which is with S, these are samples from leaves. They are not expressed in leaves, so we want to have genes which are specific for roots, because you have in roots also genes which are present, of course, in leaves. And here we separated the data to genes which are mainly regulated in fibrous roots and genes which are mainly regulated in storage roots. We were able to extract genes which are very specific expressed, whether these are in fibrous roots or in storage roots. And even, I mean, these are some names there. I don't want to go into this. On single roots, qPCR on single roots, we were clearly able to distinguish. To make the whole story, long story short, we have genes which we can associate to what kind of roots which kind of roots we have, and we have now a possibility to push with technologies, biotechnological approaches, to have earlier storage roots, to have bigger storage roots, and to have them getting even more and more shaped storage roots. I will come back to this in a later time point. The other issue we were looking at is, as I said, 300 million people in Africa are eating cassava, most of them eating only cassava, because, as I said, it's cheap and grown behind the house. Cassava is more or less starch. And we have this problem of vitamin A deficiency, which in, for example, Nigeria and Congo is a big problem. You see here, the red are really problematic regions of this problem of vitamin A deficiency. And going in farmer field, we actually found cassava which is yellowish, which could be a hint that we have vitamin A in these roots. So we thought to see whether we are able to increase the amount of this provitamin A in this cassava. Although when I was coming in the beginning, we were looking at the project, I was asking why they just don't eat tomatoes and they get their provitamin A. But these are the two problems. One is, again, they don't have the money to buy tomatoes, and B is just those are cultural. Tomatoes not something we eat in Nigeria. So here's just a measurement, the relation between starch content and provitamin A content in different varieties. You see this one is a white one, and then you have different concentrations of beta-carotene, but also different coloration of the root. This is the pathway of the metabolism. You get to up to here. Here is our product. These two products, beta-carotene and the 9-cis beta-carotene, are the components which can be converted into vitamin A in humans. All other components, which also are colored, are not useful for this case. So we were, again, of course, RNA-seq, sequencing different varieties with different concentrations. So you see the fourth three are white cassava, and then increasing concentration of beta-carotene. And we were looking where we could intervene biotechnological to improve or increase the amount of beta-carotene. Most of the publications, fruits which have beta-carotene, claim that we are on the level where you see the PSY up here. This should be the level where you should interact. If we look here, we have leaves. We're not really interested in this because beta-carotene is a part of the photosynthetic machinery. We are more interested in this thing here. And we see, actually I have to change that you see it. There are phytin synthase, PSY, and this OR, orange gene, which should actually regulate. But they are not really regulated between the white and the yellow ones. But we found this downstream NCD gene, which is clearly regulated between having beta-carotene or not. And this is down here. So we see that this gene, this enzyme, is probably pulling away the beta-carotene for abscisic acid and other hormone production. So we are about to try to knock this out and see whether this has an influence as we hope. And the third and the last topic is CMD. This is cassava mosaic disease. This is a virus disease. African cassava mosaic virus. You see two plants which are sick up there and two plants which are resistant. Breeders, by breeding activities and in crossing, were able to calculate the QTL. So they told us that we should have the gene which is responsible for resistance on chromosome 12. So we did again RNA-seq. The approach was just to see that we are taking genes which are not regulated between the two sick and between the two healthy, but regulated between the sick and healthy. And we managed to get about 257 genes which have these properties, reducing all this to the QTL, and we got one gene, which is XTH9, which is an endotransglucosylase, which had something to do with permeability in the cell wall and most probably virus movement. Here is an experiment which I mentioned before. We started first to do this experiment on Nicotiana bentamiana, which is the white mouse of the plants. And here we do a virus-induced gene silencing in bentamiana. So TRV is the virus which should do the silencing, and then we infect with the African cassava mosaic virus. If we have a control, we see high level of XTH9 and high level of virus. If we infect with the virus, Tobacco virus, with a piece of sequence of XTH9, of course, XTH9 is silenced and the virus is dramatically reduced. And now we are on the step that we can do this on cassava. We have now a virus, we can infect the plant, doesn't get sick, but it is silencing. This is an experiment first with PDS, again involved in the photosynthesis development. And if this is silenced, you have bleaching and you see clear bleaching in the leaves. And in all three cases, we will apply and see whether this is functionally correct, what we demonstrated with bioinformatics, and then see for gene editing to convert these plants into plants which are resistant, produce more vitamin, provitamin A, or have earlier rooting for higher yield. And last but not least, this is an experiment of epigenetics in cassava. While we're doing all these experiments with the roots, we found two genotypes, 117 and 693. The breeders claimed that these are duplicates according to the GBS analysis. And we found out that this one here, 117, is highly susceptible. You see the sick plant up there, and the 693 is highly resistant. But genetically almost identical, phenotypically big difference. So we sequenced the DNA methylation and did the analysis. This is just one of the example, but just the take-home message is that the 693 virus-free and the 693 infected have more or less the same DNA profile. methylation profile, and the 117 virus-free and infected is different. So we have a dramatic change. I mean, this is just one case, but we have this all over the genome. Dramatic change of the methylation profile. And the most important part is that the 117 infected becomes very similar to the 693. So the susceptible gets similar to the resistant. So that is part of what I wanted to show you, how we apply bioinformatics in Africa and start first in the farmer field.